First, an impulse travels down the motor neuron axon to stimulate the muscle fibers. Acetylcholine is released at the neuromuscular junction. This is the synapse between the motor neuron and the muscle fiber. The acetylcholine binds to receptors on the sarcolemma. This results in a stimulation of the sarcolemma. An impulse spreads across the muscle fiber and along the transverse tubes. Calcium ions are released from the sarcoplasmic reticulum and into the sarcoplasma. This increases the concentration of the calcium in the sarcoplasma. As the calcium floods into the muscle cell, it binds to the troponin. Troponin is attached to the protein tropomyosin within the actin filaments. As the calcium attaches to the troponin, it causes a change in the muscle cell that moves the tropomyosin out of the way so the cross bridges can attach and produce muscle contraction. As the tropomyosin moves out of the way, the binding sites are exposed. Muscle contraction begins when a bound ATP molecule hydrolyzes into ADP, an inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on the actin, forming a cross bridge called the power stroke. In order for the myosin to release from the actin, ATP is needed. This process then repeats itself as many times necessary or until ATP runs out. In order for a muscle to relax, acetylcholine is broken down by ACHE, which ends the stimulation of the muscle fiber membrane in the sarcolemma. Calcium ions are actively transported back into the sarcoplasmic reticulum. The tropomyosin then returns to covering the binding sites on the actin filaments. The muscle fiber remains in this position until stimulated again.